Hello dear viewers, welcome to Crown Biology classes. This is Rohit speaking, and in today's lecture I will be explaining the Ramachandran plot. So first of all, dealing with the basics. How peptide bond is formed. Peptide bond is also known as amide bond and it is formed via a dehydrolysis reaction. What happens in a dehydrolysis reaction? Removal of water molecule occurs. This is the first amino acid and this is the second amino acid. OH group from the first amino acid and H group from the second amino acid is removed in the form of water molecule. As a result of this condensation reaction, peptide bond is formed. C double bond O and H. This bond is known as peptide bond. And this process is thermodynamically unfavorable as it requires energy in the form of ATP. So it is also known as androgonic process. An unfavorable or androgonic reaction is the one in which the energy state of the product is higher than that of the reactants. That means Gibbs free energy change is greater than 0. So the peptide bond formed has partial double bond character, and how does this partial double bond character is acquired by peptide bond? It is due to resonance, and that keeps the structure in a rigid planar configuration. Because of the delocalization of electrons from the double bond oxygen to the peptide bond has partial double bond character, and that prevents free rotation around the bond. And due to the double bond character, free rotation around this bond is not possible. So the partial double bond restricts the rotation of the bond. Normal carbon nitrogen bond length is 1.45 angstrom. As a result of this partial double bond character, the peptide carbon nitrogen bond length becomes 1.33 angstrom. It reduces from 1.45 to 1.33 angstrom. Bond nearly always has trans conformation because it is more favorable than cis conformation. As you can see in the diagram, then comes cis and trans conformation of peptide bond. The peptide bond nearly always has trans conformation because it is more favorable than cis conformation. As you can see in the diagram, the trans conformation is more favorable because the alkyl group attached to the alpha carbon atoms are present on the opposite sides, but in case of cis conformation, these alkyl groups are present on the same side. These alkyl groups are bulky groups and these are responsible for steric hindrance. Steric hindrance is nothing but crowding. So trans conformation is favored in case of this. It is so because the steric hindrance between the functional group attached to C alpha atoms will be greater in the cis conformation. But in case of proline, cis conformation is more favorable. And this proline is the only amino acid found in cis conformation due to its structure. The next is torsion angles. These torsion angles are also known as dihedral angles or Ramachandran angles. So the first angle, that is between N and C alpha. The first angle between nitrogen and C alpha is known as phi. The second angle is between C alpha and carbon of carbonyl group, that is psi. The third angle, that is between carbon of carbonyl group and nitrogen, is omega. Or we can call these as phi bond, psi bond and omega bond. Torsion angles describe the rotation of polypeptide backbone around the bonds between phi, psi and omega. And this phi and psi can have any value between minus 180 degree and plus 180 degree. But within this range many values will be inhibited. And why those values will be inhibited? As a result of crowding or steric hindrance. The shape of the peptide chain can be defined by the three consecutive bond torsional angles. The first bond is NH to C alpha. Rotation around this bond is free and the torsion angle defined is phi. The second bond is C alpha to carbonyl group. Rotation around the bond is free and the torsion angle defined is psi. The third bond is carbonyl to NH, that is peptide bond. Rotation is rigid planar due to double bond character. As a result of resonance, this peptide bond acquires double bond character, and due to this double bond character, its rotation is restricted and it becomes rigid planar. And the torsion angle defined by this bond is omega. So here comes finally the introduction of the Ramachandran plot. Ramachandran plot is basically a graph introduced by G.N. Ramachandran, and it is a space filling model of peptides used to visualize energetically possible. Energetically possible means sterically permitted, not the sterically hindered. Energetically possible, that is sterically permitted values for dihedral angles. As you can see, the values of psi are given on the y-axis from minus 180 degree to plus 180 degree, and the values of phi are given on the x-axis from minus 180 degree to plus 180 degree. So a Ramachandran plot is a plot to visualize energetically allowed regions for a polypeptide backbone, torsion angles phi and psi of amino acid residues present in a protein structure. So let's discuss Ramachandran plot in more detail to have clear picture of it. G.N. Ramachandran for the very first time made a plot for which protein? Collagen protein. This is the most abundant protein in the body. Ramachandran, he worked on the permitted values of phi and psi that we have already seen. On x-axis we will take the values of phi, and on the y-axis we will take the values of psi, and he plotted a 2D map, two-dimensional map, that is called Ramachandran plot. And what does this plot basically show? It basically shows the secondary structure of the proteins. We know very well that the secondary structure of proteins consists of the alpha helix and the beta plated sheet. Allowed regions are present on the Ramachandran plot. There will be some regions that are known as allowed regions, and the regions with higher number of dots is the allowed region or low energy region. Low energy region, that means the regions are more stable. The regions where some secondary structure of proteins can be present are the allowed regions on the Ramachandran plot. The second type of regions is crowded regions. These are present around allowed regions. And the third type of regions is restricted regions, also known as forbidden regions or disallowed regions. Here no amino acid is present. No amino acid is present because of the steric hindrance. There will be more crowding. As a result of that, no amino acid will be found here. In Ramachandran plot, four quadrants are present that we have seen. Each dot on the plot shows the angles of an amino acid. So each dot has its own significance. It shows the angles of an amino acid. So this is the main diagram of the Ramachandran plot. As you can see, these are the four quadrants: first, second, third, fourth. Four quadrants are present, and each dot, these are the dots present on the Ramachandran plot. So these dots shows the angles of an amino acid. G.N. Ramachandran, he worked on the permitted value of, this is phi and this is psi. On x-axis we will take the values of phi ranging from minus 180 degree to plus 180 degree, and on y-axis we will take the values of psi ranging from minus 180 degree to plus 180 degree. And the plot basically shows what? The secondary structure of the proteins. As you can see here, collagen helix, beta plated sheet parallel is present. Here also beta plated sheet anti-parallel present. Alpha helix left-handed is present. So what is this basically the plot is showing? So the plot basically shows the secondary structure of the proteins, and there are allowed regions present on this Ramachandran plot. Those allowed regions are, see, these are the allowed regions. Where the number of dots are more, these are the allowed regions or low energy region, or the more stable region, or these are the regions where the secondary structure of proteins can be found. Crowded regions are the present around allowed regions, and the restricted regions are present on this plot where no amino acid is present. L amino acid and D amino acid. L and D means levorotatory and dextrorotatory respectively. L amino acid means which rotates the plane polarized right towards left, and D amino acid means which rotates the plane polarized right towards right. And in bacteria, both L-chiral and D-chiral amino acids are present. So what does this term chiral means? When a carbon atom is bound to four unique groups, it creates a chiral center that is also known as stereocenter. In bacteria, both kinds of amino acids are present, but in humans only L-chiral amino acids are found. So this is a picture depicting Ramachandran plot, L-chiral and D-chiral amino acids. In L-chiral amino acids, In first quadrant, beta sheet is present. In second quadrant, left-handed alpha helix is present. Third quadrant is vacant. In fourth quadrant, right-handed alpha helix is present. And what happens in D chiral? Opposite situation of L chiral is present in D chiral. In second quadrant, right-handed alpha helix is present. In third quadrant, beta sheet is present, and in fourth quadrant, left-handed alpha helix is present. Opposite to this quadrant is right-handed alpha helix. So accordingly, this left-handed alpha helix will be found in fourth quadrant in D chiral, whereas it is present in second quadrant in L chiral. Beta sheet is present in second quadrant in D chiral, whereas it is present in first quadrant in L chiral. And right-handed alpha helix is present in second quadrant in D chiral, whereas it is present in fourth quadrant in L chiral. There are few exceptions of the Ramachandran plot. The cyclic side chain of proline limits its range of phi values to angles of around minus 60 degree, making it not surprisingly the most conformationally restricted amino acid residue. As you can see in the structure of proline, there is a cyclic side chain. So this cyclic side chain is causing the maximum restrictions. Second one is glycine. This is the only residue without a beta carbon atom. It is much less sterically hindered than other amino acid residues. It is much less crowded. As a result of that, it will be found on the Ramachandran plot maximum times. Hence, its permissible values of phi and psi covers a larger area of the Ramachandran diagram, and at glycine residues, polypeptide chains often assume conformation that are forbidden to other residues. So it will be occupying the maximum space on the Ramachandran plot because it is very less sterically hindered. Thank you for watching.